Many people, including so-called authorities who promote a plant-based diet, claim that the structure and function of our digestive tract is very similar to chimpanzees and other great apes. However, this illustration, drawn to scale, depicts the very significant difference between our gut and those of the great apes, including chimpanzees and gorillas. Evidently, a healthy human has a much smaller gut. The great apes have big guts and small brains, and we have big brains and small guts. In fact, our brain is about 3 times larger, and our gut mass is about 40% smaller than expected for a primate of our size. In primates and all other mammals, both brain and gut mass are related to diet. Species that specialize in feeding on low energy density plant foods have relatively large, complex guts with either foregut or hindgut fermentation vats, along with small brains. And species that specialize in feeding on high energy density animal foods have relatively larger brains but relatively smaller and simpler guts without capacious fermentation vats. We fit into the latter category. Comparing our guts to those of the chimpanzee or gorilla, we find markedly different proportions devoted to the main compartments. While we all have a similar proportion of the gut devoted to the stomach, we devote much more of our gut, 62%, to the small intestine, where enzymatic digestion and assimilation take place. Whereas in the chimps and gorillas, the colon is the largest portion, about 52% of the gut. Moreover, the great apes all have well-developed cecums, whereas we do not. Instead, we have the vestigial vermiform appendix. Whereas the cecum performs vital digestive functions for the great apes, our appendix has no vital digestive function, as proven by the fact that many people have the appendix removed with no loss of digestive ability. This is because the chimps and gorillas are designed by nature for hindgut fermentation of a high fiber diet, whereas we are designed by nature to rely on enzymatic digestion of foods in the small intestine. It bears noting here that our pancreas produces no enzymes for digestion of fiber, which leads to the conclusion that nature did not design us for digesting plants, all of which are in nature primarily composed of fiber. This graph drives home the point. We have the majority of our gut volume in the small intestine, almost no cecum, and comparatively little in the colon. All the other listed primates, including chimps and gorillas, have the majority of gut volume in the colon. They are all hindgut fermenters, and we are not. There exists an absolute difference between our intestines and those of the great apes. Chimpanzees have a longer appendix, a much lower number of loops in the small intestine, and increased hostrations, and therefore much more surface area in the colon as compared to humans. This gut reduction in humans signals a significant difference in natural diet. Humans have eaten animal tissues, marrow fat, brain matter, and muscle since at least 2.5 million years ago. These tissues are energy dense and digestible without microbes, making it unnecessary for us to have a capacious, metabolically expensive gut like found in chimps and other great apes. Some people assert that we are herbivores because we have a greater ratio of gut, that's total intestine, length to body or head to tail length than found in hypercarnivores like the dog or the cat. As recorded in my previous book, Powered by Plants, I at one time believed this. However, I have since realized that this argument fails for two reasons. First, a mathematical point. Length alone does not determine volume or surface area. The intestine is a cylinder. Two cylinders can have similar length but dramatically different volume because you must account for diameter to determine total surface area and volume. These two cylinders have the same length of 20 centimeters, but the upper one with the larger diameter has a surface area more than double and a volume about 4 times that of the one below. Hence, two species could have similar bowel lengths relative to body length, yet one could have dramatically greater bowel volume and surface area than the other, if the bowel was in a larger diameter. Bowel length alone can't tell us anything about bowel volume or surface area, but it is in volume and surface area, not just length relative to body mass, that herbivores differ from carnivores. To reiterate, length alone is immaterial. You must also account for diameter, surface area, and volume. Assuming similar energy requirements, the difference between an herbivore gut and a carnivore gut is the herbivore will have greater surface area and greater volume due to the need to process a very bulky diet. The mere fact that our intestines are, relative to body length, somewhat longer than those in cats and dogs does not prove greater surface area and volume relative to energy requirements. Regardless of length, the fact that our gut has much less volume and much less surface area than that of the herbivorous apes is sufficient to indicate that our gut is designed for a low bulk, high energy density diet. More importantly, focusing only on ratio of gut length to body length ignores the differences in relative energy needs between different carnivorous species. We have the largest, most energy expensive brains of all species. Relatively speaking, our brain is proportionately twice the size of the dolphin, 3.3 times the size of the chimp, 10 times the size of a lion's or gorillas, and 100 times the size of a horse's. Our brain consumes about 25% of our daily total energy expenditure. To support the high energy requirement of our extraordinarily large brains, we need to extract far more energy from food than any other species, including any other carnivore. Consequently, at the same body size, we absolutely need more volume or length in the small intestine than other carnivores. Thus, the fact that we have a ratio of body length to bowel length somewhat greater than other carnivores absolutely does not indicate that we are designed by nature for a plant-based diet. Those who think it does are ignoring the enormous difference in brain-to-body ratio and consequent energy needs between humans and other species of similar body size, including both the herbivorous great apes and the carnivorous canines and felines. Now let's take a look at how our stomach acidity compares to that of hindgut fermenters and omnivorous primates. Since maintaining a highly acidic stomach is costly, we expect to find it only in species where it is adaptive. One of the main functions of the stomach is to act as a filter that destroys foodborne pathogens in an acid bath. Natural herbivores have low stomach acidity because plants rarely carry bacteria that are pathogenic to mammals, and plants are best digested in an alkaline environment favorable to microbial growth. In fact, herbivores depend upon large microbial populations for digestion of fiber. A very acid stomach would destroy these microbes, making it impossible for the herbivore to digest its food. In short, a highly acid stomach is maladaptive for an animal designed to subsist on plants. In contrast, an acidic stomach is highly adaptive for a meat eater because it will destroy pathogenic bacteria in the meat that could cause illness or death, and because acid and pepsin are highly effective at dissolving meat and fat. Thus, stomach acidity is a predictor of an animal's natural diet. The more acid the stomach, the better adapted the animal is to eat a meat-based diet. Obligate carnivores will have the highest stomach acidity. Further, since bacteria are specialized for infecting different classes of animals, for example birds, mammals, etc., a carnivore would have the highest risk of bacterial infection if it eats meat of more closely related or the same class of animal. For example, a bird eating a bird or a mammal eating a mammal will have the highest risk of infection compared to a bird eating an insect or a worm or a mammal eating an insect. Hence, stomach acidity should be highest in animals that are designed by nature to eat meat from their own class or closely related classes of animals. So here we have the stomach acidity of some of the species representative of different types of dietary adaptation. Data for chimps and gorillas was not available, but they are hindgut fermenters dependent on microbes, so it is reasonable to expect that they have stomach acidity similar to rhinos and elephants, which have a pH around 3.3 in the stomach. The omnivorous primates, the baboon and Skypes monkey, have stomach acidity similar to the hindgut fermenters. In stark contrast, humans have a stomach pH of 1.5. Let me repeat, our stomach acidity is pH 1.5. Understand that pH is a logarithmic scale, so a pH of 1.5 is about 63 times more acidic than a pH of 3.3. Once more, our stomach acidity is about 63 times greater than that of omnivorous primates or hindgut fermenting mammals. In fact, our stomach is more acidic than the domestic dog or cat, both obligate hypercarnivores. To be precise, according to this data, our stomach acidity is 100 times greater than the cat and 1,000 times greater than the domestic dog, although I suspect these numbers for the cat and dog come from somewhat ill domestic cats and dogs fed commercial pet foods. Our stomach acidity is most similar to that found in the falcons, hawks, gulls, and kestrels, all either scavengers or generalist carnivores. Recall that maintaining a highly acidic stomach is very costly and maladaptive for herbivores. Nature would not have given us this highly acidic stomach if we were to eat a plant-based diet, because it is maladaptive for an herbivore. It makes digesting plants more difficult by killing the microbes that help herbivores to digest plants. This strongly supports the conclusion that we are designed by nature for a highly carnivorous diet, either as a generalist carnivore or scavenger. Yes, you will notice these authors labeled humans omnivores. Almost everyone does, because that's what they're taught to do, and because, as a matter of fact, humans do eat both plants and animals. But what people do as a matter of fact does not tell us what people are designed by nature to do. For example, as a matter of fact, people smoke tobacco, drink alcohol, snort cocaine, shoot up heroin, and drink Coca-Cola. But this does not prove that people are designed by nature to do any of these things. Moreover, the vast majority of carnivorous species are actually omnivorous to some extent. As I explained in my video discussing the scientific definition of carnivore, linked above, most carnivores, including big cats, wolves, polar bears, and crocodiles, voluntarily eat some plants. Consequently, biologists further classify carnivores into subcategories: hypocarnivores that get less than 30% of their food from animals, mesocarnivores that get 50 to 70% of their food from animals, and hypercarnivores that get more than 70% of their food from animals. Further, since an omnivore by definition eats both plants and meat, and the word carnivore simply means meat eater, all omnivores are logically, by definition, carnivores. If you think calling humans omnivorous is not calling them carnivorous, you have a poor grasp of logic. Also, as you have seen, most so-called herbivores eat some animal matter. In other words, practically all animals are omnivores. So labeling an animal an omnivore is biologically practically meaningless and a cop-out, a refusal to identify the animal's biological specialization. The question is, what is the animal designed to specialize in eating? This stomach acidity data provides strong evidence that we humans are designed by nature to specialize in eating meat of mammals, birds, and other creatures that could transmit to us pathogenic bacteria. Gorillas and chimps have similar diets. Both are so-called omnivores, but they specialize in eating fruits and vegetables. The Western lowland gorilla's diet is about 67% fruit from 100 different species, and 3% termites and caterpillars. And the balance is composed of other high-fiber plant matter. These foods contain about 90% water, and the wild fruits that gorillas eat contain more dietary fiber than leaves and vines, unlike the high-sugar man-made fruits modified by agriculture. Direct analysis of the gorilla's foods suggests a very low-energy, low-fat diet providing 8.3 kilocalories per kilogram, or 83 kilocalories per 100 grams of dry weight. Before accounting for hindgut fermentation, the gorilla's diet consists of 57% of energy from protein, because leaves are very high in protein, 37% from enzymatically digestible carbohydrates such as starch and sugar, and 6% of energy from fat present naturally in fruits and vegetables. At only 8.3 kilocalories per kilogram of food, a gorilla would have to consume 361 kilograms of raw food daily to get a young man's daily requirement of 3,000 kilocalories. This is obviously a prodigious amount of food, impossible to consume in a day, even for a gorilla. Nature found a solution to this problem in hindgut fermentation. The gorilla's large cecum and colon are full of microbes that can turn all that otherwise useless fiber into fatty acids. When you account for the energy a gorilla gets from short-chain saturated fats produced in its colon by microbial fermentation of fibers, the energy density of the diet more than doubles from 83 kilocalories per 100 gram to 194 kilocalories per 100 gram dry weight. Moreover, it turns out that the gorilla is not on a low-fat diet at all. In fact, it actually gets 57% of its energy from saturated fats, and 60% of its energy from total fats, including those present naturally in fruits and vegetables. and only 16% of its energy from starches and sugars. The gorilla eats a low carbohydrate, high fat diet. The authors of this report include Popovich, Jenkins, and Kendall, all of whom advocate a plant-based diet because they believe we should eat like other primates. In a few minutes, I will show results from one of their experiments in which they put humans on a vegan simian diet composed exclusively of fruits, vegetables, and nuts. However, these authors recognize that evidence indicates that fiber fermentation in the human colon only contributes 2 to 9% to total energy requirements, compared to, according to their own calculations, 57% in the gorilla. In light of this fact, how can they imagine that we are designed to eat the same kind of diet as a gorilla? To me, this shows how the mind can ignore pertinent evidence refuting its beliefs when it is unwilling or unable to reconsider those beliefs in light of such evidence. Now, Popovich et al. Have shown very clearly that the capacious colon is an absolutely vital organ for the gorilla or chimpanzee because it provides the animal with most of the energy it needs to live. Relatively speaking, the colons of these great apes contribute 10 to 15 times more energy to their energy needs than our colon contributes to our energy needs. To reiterate, these great apes absolutely cannot live without their colons or the microbes in those colons, but we have strong evidence that colonic fermentation of fiber is of no more nutritional importance to us, humans, than it is to cats and dogs, both archetypical hypercarnivores. In fact, in respect of deriving energy from fiber, our colon functions much more like that of dogs and cats than like that of a chimpanzee or gorilla. According to this manual on dog and cat nutrition by the National Research Council, a dog fed fibers derives about 7% of its total energy from saturated fatty acids produced by fermentation in the colon, and it is probable that the cat does the same. To reiterate, Popovich et al. Stated that the human colon can contribute only 2 to 9% of total energy, similar to, or perhaps even less than, what is found in dogs and cats. The claim that our gut functions like that of a great ape lacks factual basis and represents ideology, not biology. As I mentioned, Jenkins, Kendall, and Popovich teamed up to study the effects of a vegan great ape diet on the human colon function. They fed human volunteers a simian diet consisting of fruits, vegetables, and nuts, which provided about 150 grams of dietary fiber daily. In Table 5 of this report, they report that the people eating the vegetable diet excreted large amounts of short-chain fatty acids in their feces. These are the same saturated short-chain fatty acids that gorillas depend on for 57% of their energy needs. Whereas gorillas absorb these short-chain fatty acids, the people eating the very high fiber simian diet had a total fecal excretion of short-chain fatty acids that was about 5 times that of people eating a diet containing only about 25 grams of fiber per day. Yes, more fermentation was taking place in the colons of those on the high fiber diet. The flora were having a party, but most of the fats produced by this very high fiber intake were of no energy value to the people eating that diet because they ended up excreted in their copious feces. Remember, this data is provided by people who are themselves ideologically committed to the idea that we should be vegans or nearly vegan, eating diets similar to the great apes. Yet these data fairly clearly demonstrate that, unlike gorillas, humans do not efficiently absorb short-chain fatty acids from the colon. One likely reason being that our colon is too short and compact to allow adequate time and surface area for absorption, unlike the capacious colons of chimps and gorillas. In any case, these advocates of plant-based diets have themselves shown that humans do not assimilate short-chain fatty acids from the colon the way that great apes do. Simply put, unlike the great apes, we are not hindgut fermenters, therefore not designed by nature for a high-fiber diet. Here is the most decisive evidence indicating that colonic fermentation of fiber is of no significant nutritional importance to humans. According to the journal Ostomy Wound Management, between 450,000 and 800,000 people in the U.S. have some type of ostomy, and about 36% of those are colostomies. That means somewhere between 162,000 and 288,000 people, or about 250,000, are living without a colon. Hence, even if we can derive 2 to 9% of our energy from fats absorbed from the colon when consuming a plant-based diet, it is of little importance to us. We all can absolutely live without the colon and therefore without fermentation of fiber in our guts. Similarly, we can sterilize our colons with antibiotics and still obtain adequate energy from our foods without the help of microbial fermentation. In this respect, we are the same as cats, dogs, and other hypercarnivores. No carnivore needs colonic microbes to ferment foods in order to survive. In stark contrast, this would be impossible for a gorilla or chimpanzee on its natural diet, because these animals depend on colonic fermentation of fiber by microbes to obtain about 60% of their total energy requirements. For them, a colostomy or sterilization of the colon would mean certain death from starvation. In summary, one of the main differences between a natural carnivore, that's an animal designed by nature to specialize in meat eating, and, on the other hand, a natural herbivore, that's an animal designed by nature to specialize in eating plants, lies in the relative dependence on microbial fermentation for survival. Simply put, an obligate herbivore cannot survive without its fermentation vats full of microbes to digest fiber, whereas a natural obligate carnivore has no need for fermentation and can survive without gut microbes, but in its natural habitat must consume animal meat and fat to satisfy its nutrition requirements. We humans belong to the latter category.